In this finale to the year, we're going to be combining a number of topics, but with a general theme. So we've just finished looking at cell reproduction, and we've looked at mitosis, and now we're going to look at how genetics plays into how we inherit certain properties, certain characteristics, what chromosomes, how they're involved with that. And yeah, so let's just get started. So the first thing we want to just remind ourselves of is we talked about mitosis. When we looked at mitosis, we were looking at somatic cells, cells which are just body cells, could be a skin cell or any type of cell that is going to replace itself or go through a process so it can repair some damage, or maybe it has to do with growth. But when we see cells like that, what we're looking at are cells which have a full set, actually two sets of chromosomes. One was that you received through the father, one through the mother, and we say that is a diploid cell. So for us, diploid means that there are 23 pairs of chromosomes. We also can use the notation 2N, which also indicates diploid. So we've looked at the cell cycle. You have a pretty good handle on certain things that regulate the cell cycle along the way. We know that in G1, this is representing a certain amount of DNA for all that particular cell or cells in the body. And then we know that in the S phase, we make a copy of that DNA. So and when we look at a cell, and if we took out all the chromosomes and we put them together, we'd see that they're all on some type of a matched set, which is called a karyotype, meaning that chromosome set number 1, you received one copy of that from your mother, one from your father, but on chromosome pair number 1, we're going to have very particular information. That's that same thing for chromosome 2, 3, 4, 5, etc. You can see that they vary in size and shape. So that's kind of significant. It also helps us to figure out which ones are the pair that fit together. So here's mitosis. You remember this. So here's the 2N, right? That idea that there's a copy of the chromosomes that your mother contributed and those from your father. So we have this 2N condition, diploid condition. We go through the whole phases, all the phases of mitosis, till we get these two daughter cells, which also are 2N, because we already copied the DNA back in the S phase. Now we're just dividing it so that each of the cells has a full complement or full set of that. If I'm a bacterial cell or a prokaryotic cell, it's so much easier. I have no nucleus, right? So all I'm going to do is make a copy of this big loop of DNA. And after having done that, we'll just separate the copy from the original, and then we're going to divide in two. We're going to do what's called binary fission. But producing sex cells, which are very specialized cells, means that we're going to create some cells which only have 1/2 of the chromosomes as in the average body cell or somatic cell. And what that means is they're going to have one set of chromosomes. So when we look back at that earlier picture of all the chromosomes, they would only have 1 out of the 2 for each one of those pairs. So the haploid condition is this 1N. And meiosis is what we might think of it as a double division. So once we've copied the genetic material, the cell is going to divide like it does in mitosis, but then it's going to divide again, and those chromosome numbers are going to get halved again. We'll see that here. So meiosis there's 2 rounds of meiosis. We start with this parent cell, which is a specialized cell. This would be found in humans, it'd be found in ovaries, In males, it'd be found in the testes. And so these aren't average cells, these are specialized cells. And they are going to do similar to mitosis, the kind of the same idea. We're just going to make copies of all the chromosomes, and then we're going to split ultimately into some daughter cells. But this process is going to be dividing twice. So this would be the end of telophase 1, where we'd have these two cells. But then we're going to go on and do kind of the same thing again, except for there's no copying of genetic information. So when we split again, we're going to end up in the second telophase with these 4 cells here. And so telophase 1, so there'll be a prophase, metaphase, anaphase, telophase 1, and they'll be the same thing, prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. So meiosis is going to produce these sex cells, which reduces our chromosome numbers from diploid to haploid, which will ensure that every cell has one complete set of chromosomes. And ultimately, another major function of this process, which we'll get into some detail on, is it's going to generate diversity among the products. So in other words, it's going to lead to a tremendous amount of variation that we're going to see within any given species, or even within a given family. So here we go. Let's go through this once again. So meiosis 1, the first round of it, is where we get the homologous chromosomes. Each of them is going to make a copy. So when we say homologous here, that means that this is what you got, one copy of the green maybe from your mom and the pink from your dad. Might as well say that, right? And if you had these in the original cell, then each one of these are paired. These are homologous because they have basically the same type of information on them. So this could represent chromosome pair number 1 here. But each one of these are also going to make a copy, right? So here's our sister chromatids for each one. And then the sisters are going to stay together, but we're going to see something interesting. So let's go to this. So in meiosis 1, here, this is kind of a photographic view, but let's look at the drawings here below. So we can see early prophase, chromosomes become visible, the centrosomes or the centrioles start to migrate, nuclear membrane is going to break down. And then what's really fascinating in this meiosis is that the pairs of homologous chromosomes, the ones we just saw a minute ago, like back here, I'm going to flip back, this right here. So here we have the homologous chromosome pairs with their copies. So they're going to stay together in meiosis 1. It won't be random, they'll stay together. And they're going to form together something called a tetrad, which is the 4 of them right next door to each other. And when that happens, they're going to be able to, the middle ones, the ones that are closer in contact, are actually going to exchange bits of information. Some of the genes, in other words, there are going to be some exchanging that goes on. Metaphase 1 is going to look much like mitosis. They're all lined up in the middle. But notice here on anaphase, when the chromosomes are pulled to opposite poles, the sisters here are staying together. So the sisters, with a little bit of change in them, a little variation, because they did this crossover. And then we're going to end up here in the end, a telophase, with these two cells, which have still they still have the sisters, one set of chromosomes, but also with their sisters together here. And so what you'll see is, we already said this a little bit, this idea that at the end of meiosis 1, we're going to have 2 new nuclei, and they're going to have a half of the original chromosome numbers, because what they have are the sisters are still together. Now, meiosis 2, there is no more replication of DNA. So it's in meiosis 2 that the sisters are going to separate, and we're going to end up with 4 haploid cells, which will have some differences in them. They won't be genetically identical. And so meiosis 2, we go losing, the nuclear envelope disappears. We have the sister chromatids are now condensing so we can see them. Then we get that they line up in the middle, so you can see that here. Then they separate during anaphase. And then during telophase, we form new nuclei, or nuclear membranes. And then we divide again. So when you end up at the end of meiosis 2 with 4 individual gametes, or sex cells, each with only 1/2 of the chromosome number of the original cell. That crossing over is kind of important, so we're going to take a look at this again. So the genetic material gets shuffled during meiosis. Crossing over is when the homologous pairs, meaning the original pair plus their sister chromatids, line up together, form something called a tetrad. Tetra means 4, right? So we can see that here. So the homologous is the red originally, and the blue, these are their homologous pairs with their sisters, with their copies, are lined up. And then we have some exchange of DNA in these areas, which actually, on this part in here where there's a crossing, that's called a chiasmata, because a chiasm means to cross. And you can see that here. The centromeres, remember, are just still holding them together. This is where the chromosomes are actually exchanging some of that genetic information. And when it's exchanged now between the things which are the non-sisters, because it wouldn't make sense for a sister to exchange, it's the same information. So you can have this information being exchanged between all of these different chromosomes, and that results in something called recombinant DNA, or recombinant chromosomes. And what we'll see coming up here, here's the homologous chromosomes together here with their sisters. They cross over. At that crossing point, it's a chiasma or chiasmata. At the end of that, they go back, and you have some little bit of recombination right here between these two. And then that is kind of the end. And actually, that's probably a good stopping point here. So let me stop here, and then we'll get into just a little bit more about how this all works.